I think we've you know, kind of stabilized. It's good to see so many familiar faces. And I see some very new ones. I think I may have seen some fairly young children, perhaps related to our speaker. But
you can't start too early.
That's right. That's right. So for those of you who don't remember me, I'm Mike Ogilvy, and I took over organizing these Saturday science lectures brought to you by the physics department at Washington University a couple of years ago. I can't tell you how pleased I am to see you all. So this series is a chance for you all to hear from some of the newer members of the physics department. You know, not too new, but some of the shininess is not quite worn off. You know, I'm excited to be here today with you to hear what they have to say. We are already working on the fall series. We don't quite know how we'll be able to present it because we actually don't quite know how we're going to do classes in the fall. But, you know, it's being worked on even as we speak. So, you know, you'll all be notified in the fall if you signed up for our email to find out about those talks. So,
excuse me, may I say something? This is important. Those of you in the WashU Physics Faculty all know Sonia Bahar at UMSL. Mm-hmm. And you also know that UMSL is trying to destroy that program. And so for those of us who love science and who love physics, if you have any connection to UMSL or provosts or people where you can speak out, I know Sonia and her colleagues deserve it. So I just wanted to say that loud and clear because I'll be seeing her later today and I'm going to tell her that
yes. The physics department already here at Washington University already circulated a joint letter signed by most of the faculty here supporting the UMSL physics department as well. Yeah, that is an important issue in our community. So, you know, I want to turn it over to Professor Mukherji as soon as I can, but I just thought I would tell you that he came to us with a distinguished background. Like me, he got his undergraduate degree from MIT, but then he went on to get his doctorate, his PhD in biomedical engineering from the Harvard MIT Division of Health Science and Technology, and came to Washington University after a postdoctoral fellowship at the Center for Systems Biology at Harvard. And of course, he is a biophysicist. And today he's going to tell us about his work on cellular self-assembly.
All right, thank you, Michael. Always nice to have shared Boston roots. Two of the three little faces on the screen you see, they're native Bostonians, so they came with me when we moved over here. All right, so let me go ahead and get started. Before I keep going, can everyone see this? I know that there's all, okay, I'm seeing some thumbs up, great.
I did it.
So, yeah, so first of all, thank you for coming. It's really nice to be able to step a little bit outside the sort of day-to-day drudgery of you know, lab meetings and looking at raw data, and take a step back and actually think a little bit more broadly about the context in which we do our work. So just to give you a little bit of background of where we're coming from, our lab really sits at the interface of physics and cell biology. And whereas a traditional cell biology lab might worry about, you see this sort of little toy cell I made. This is actually sitting on a pancake griddle, just full disclosure. Whereas a traditional cell biology lab might look and try to sort of identify what these green and blue and yellow little beads are, and maybe something about which other beads they talk to. What we're really interested in is sort of deciphering general principles by which how precisely does the cell even organize these beads? How much control can these systems exert on themselves, and what are the sort of principles by which it regulates the organization of matter and energy within itself? That's kind of where we're coming from. Slightly more abstract perspective than your traditional cell biology lab. So back in the late 1950s and early 1960s, the French microbiologists François Jacob and Jacques Monod revolutionized our understanding of how living systems work when they discovered that in response to their environments, cells will actually have dedicated systems to turn on and off little bits of their DNA called genes in order to make new proteins that will be able to handle the environmental changes that they're experiencing. And so in the course of that work, which was awarded the Nobel Prize in 1965, François Jacob sort of coined this very pithy phrase, which is that the dream of every cell is to become two cells. And this sort of primal, urge for living systems to replicate themselves is almost definitional to our idea of what life even is. So our goal is to try to understand basically the organizational principles that underlies this dream that cells have of becoming two cells. And so if you think about it, the process itself, it doesn't seem like it has to be all that complicated, right? So this is a brass sculpture of a budding yeast cell made by my colleague at the University of Chicago, Alan Drummond, who's a biochemist, actually, by day, not a sculptor. If you look at it, you sort of see, okay, well, you've got like a balloon-looking thing. It's sort of this glob, and from that glob is pinching off another little glob. And I mean, it doesn't look like it should be all that hard, right? You just sort of take this thing and maybe pinch off a little bit of that, and you get two cells. What's the big deal, right? But of course, if you actually look a little bit under the hood of what a cell is actually like, you see that the dream of every cell is sure to become two cells, but that dream is pretty complicated. And if you look inside the cell, it's not just a random grab bag of molecules diffusing around in this tiny space. It's actually a highly intricately structured object that is made up of all kinds of heterogeneous components. Things are moving around. They're small, so they're still subject to all kinds of fluctuations and noise from the fact that they live in a hot environment. So things are diffusing around really rapidly. And so, even when you look at this sort of first layer below the skin of the cell, things already look a little bit complicated. So this dream looks already a little hard to achieve. Okay, but it gets worse, actually, because if you zoom in even more from one layer down, you see that the actual chemical reactions depicted here in this chart are even more complicated than the sort of structural features we saw just one layer below the surface of the cell. What this chart is a chart of the chemical reactions that occur in cells by which cells take glucose, so simple table sugar, stuff that we eat all the time, and transform this very simple molecule into all the various complicated biomolecules that the cell needs. For example, nucleotides that make up our DNA, or lipid molecules that make up our cell membranes, or the protein molecules the protein molecules that make up most of our cells. And as you can see, that this network of reactions by which cells are able to sort of generate this magnificent diversity of molecules from this very, very simple precursor is very complicated. Okay, but it actually gets even worse, because if we zoom in on even one of those little pieces of the reaction network that I just showed you, every single one of the edges between two of the dots represents an enzyme, an actual protein machine that takes one kind of molecule and converts it into another. So let's look at this edge right there. So this is in a part of the metabolic reaction network that involves synthesis of these sort of cell surface, cell wall components. And what you see is that since every single one of those edges, those lines between dots, is a protein, it has to be made by the cell itself, right? And the way proteins are made in the cell is the cell reads out the DNA, it converts it to this molecule called messenger RNA, which is something I think all of us actually know a lot about now because of the miracles of the messenger RNA vaccines that are being pumped into us millions of times a day. And those messenger RNAs are further decoded into the actual proteins that are the enzymes that are in this chart. Now, in that process, though, of what's called gene expression, by which the DNA information is converted to proteins, that's a physical process too. And if you actually image the number of molecules inside cells that correspond, correspond to each of these little enzymes, you actually see that different cells, even though they're genetically identical, these two cells shown here are twins, they're clones, and they're exposed to the exact same environment, so they have no difference in temperature or sugar content, nothing. They will actually produce different amounts of these proteins, as indicated by the different number of, for example, green dots and red dots that you see on correspond to the two cells you see here. So what we're left with is this sort of, I find, troubling realization that the building blocks with which we ourselves are made, right? We're made up of cells just like any other living organism. These building blocks are made up of very unreliable parts. They're complicated. They're subject to all kinds of random forces that make it so that two cells, even if they're identical, can be quite different from each other. And so we have this huge puzzle on our hand that really is a defining question in modern biology and modern biophysics, which is, how does this biophysical order that we see on the outside, right? We're not, when you're sitting around, you don't all of a sudden grow a third arm or start melting away or something. We are pretty stable, more or less. So the question is, how does that order at these larger length scales, like our own daily life length scales, emerge from this noisy, nonlinear, highly interconnected and complicated network of reactions? That's the big question that people who work in my field are trying to answer. So so now I have to sort of take a step back and give you the real answer, my dirty little secret, which is we don't know. We don't know at all where this order comes from. Okay, we have some ideas, and if anyone tells you they know, either put them into touch with me because I want to know, or just sort of dismiss them because they don't know. But in trying to get at the answers to these questions, I'm going to try to take you through two different lenses that we've been looking through to try and address this question. And this is work from largely my own group. And so, there's sort of two scales that we're going to look at this problem. So one scale is we can look at how it is that cells can assemble structures within themselves individually. So we're just going to look at single structures at a time and see whether or not we can uncover some principles that govern the assembly processes of those structures, so biophysical principles. And the idea in this thrust of work is maybe we can find out that cells sort of cleverly actually regulate the processes that underlie that assembly the assembly itself, to see if we can't uncover whether the cellular cleverness underlies this order or not. Okay. Another, and so, yeah, so, right, so the idea here is we're watching this kid blow this balloon up, and we're going to focus on one balloon at a time. We're just going to look at single species of balloon. Another thrust of work in my group is about how the self-assembly processes for individual structures within the cell are coordinated with each other to promote the overall growth of the system as a whole. And I'll just touch a little bit on this at the end. This is, I'll show you some unpublished work that we've compiled. And this is really the centerpiece of what my group is focusing on right now. And the idea here is you have a balloon within a balloon, and how is it that you coordinate the growth of those internal balloons inside the overall balloon? Okay, so let's start with the first set of observations we have. So in order to actually do experimental work in this area, to actually have data to work with and test our ideas against, we have to pick some kind of system that we want to study. And in our case, the system we study is the eukaryotic cell. So the living universe is sort of divided into three large categories. One are what are called prokaryotes. Those are things that like bacteria. So we usually think of them as infectious or a nuisance, but actually they make up most of life. They're actually most of the biosphere is prokaryotes. There's a sort of intermediate domain of life called archaea that we actually don't know very much about at all. We're still learning a lot about it. And then there's these guys, the eukaryotic cells, which the hallmark of the eukaryotic cell, and we, for example, are made up of eukaryotic cells ourselves. The hallmark of the eukaryotic cell is that it's made up of these defined components that themselves are bound by membranous structures. And so my student Ali Ara made this very nice cartoon of a eukaryotic cell depicting some of the structures. So you'll see some of these pop in and out of the story throughout the talk. But one way I like to think about the eukaryotic cell, because it's this sort of structure within a structure, is it's almost like a little town or a little city, right? So the different membrane-bound compartments within that town, which are called organelles, they sort of perform different kinds of functions for that cell. So for example, in this little cartoon, you can see that there's this thing called the nucleus in the middle. Maybe I can even try my laser pointer. Usually when I try this, things crash, but okay. So right in the middle there, you have the town hall, which is the nucleus. The nucleus is the structure of the cell that contains our DNA. So this is sort of like the place where all the information is stored and from which instructions are sent out. Over here, you have the post office, right? And in the eukaryotic cell, the post office is called the Golgi complex. So this is where proteins come and they get recognized by enzymes inside this post office. They get modified appropriately, and then they get sent off to all the various destinations inside the cell. You have factories, which are ribosomes. These are the little pieces that make proteins. You have the town dump here, lysosomes. That's sort of the garbage depot of the cell. That's where cells go, that's where cells send proteins to get hydrolyzed, meaning broken down into their component amino acids for recycling for later use. And so on and so forth. I mean, I can march through this whole thing. You have down here an important one, the power station, which is the mitochondria. And And you have other things like you have storage depots for fuel, the lipid droplets. You have places where fuel is burned called peroxisomes. And there's a whole zoo of these things, is long story short, okay? And the question that we have is so if we take this, we can actually do more than just draw cartoons though, right? We can actually, with our microscopes, actually image all these different components inside individual cells so that we can actually take quantitative data from them and try and understand the answer to questions like I posed before about how do cells generate order. So I'll give you a little example of the kind of data we work with. So what you have shown here is a micrograph of budding yeast cells. So each one of these little collections of fluorescence, not the little individual dots, but the sort of the circles that sort of delimit the dots that I'm drawing. You have individual cells, and in this cell, we have genetically engineered these cells so that a particular organelle inside them will light up, and we can image that. In this case, it's the fat storage organelle called the lipid droplet. Now, what we can do is we can, for example, focus in on one of these cells in particular and blow it up, and we can look very carefully at the structures that the cell makes corresponding to these lipid droplets. In particular, what we can do is we can take out our teeny tiny rulers, right, and literally go in and measure how big each of these structures is, right? Because that's a measure of that's a kind of order that the cells generate out of their building blocks, right? Is they create these structures and presumably they try to control the sizes of these structures. So once we have this kind of data in hand, we can ask ourselves a question. How precisely can cells control the assembly process of these organelles? So in order to understand, so I want to emphasize this word precisely, right? Because this is the sort of issue that physicists really care about, right? We want to understand not just sort of the genes and the proteins that go on to making these structures. We want to understand something about quantitatively, how does the cell exert control on these structures? So to understand this issue of precision, though, we need to have some background knowledge of how to even think about what precisely means in this context. So in order to do that, let's do a little exercise. So when I give this talk in person, usually at this point, I ask for volunteers to come up to the board and do a little thing with me. We can't do that. You'll see that this exercise in particular is particularly no-no right now. So let's go back to that little boy who I randomly found on the internet and blowing a balloon. Okay. And let's imagine that what we want to do in order to understand how cells control growth is we want to see how it is that this boy is able to blow up a balloon to different sizes. So you can have a small size balloon, you can have kind of a medium size balloon, or you can have a large size balloon. Now, the rule in the game that I'm going to impose on this little boy right now is that the boy can only blow into the balloon in discrete units. So it's 1 unit of air that he's allowed to puff into the balloon. In order to achieve different sizes, all the boy is allowed to do is change the rate at which he puffs into the balloon. So you can't change the size of the puffs, just the speed with which he's puffing. Okay. The reason we impose that rule is because that's how we think assembly works. It sort of works 1 molecule being added at a time. Okay, so if we do that, let's imagine we plot the size of the balloon as a function of time as the boy is trying to blow the balloon up to these different sizes. Okay, So for a small balloon, you can imagine if I mark in red each moment the boy actually puffs into the balloon, he'll puff once and he'll puff maybe again, just to, air is constantly leaking out of these balloons, right? You know how balloons work. So he puffs a couple more times. And the idea is that every time he puffs into the balloon will grow a little bit. And in between the puffs, the balloon will shrink a little bit. And in order to keep a steady size of the balloon, he's got to match the rate at which he's puffing with the rate at which it's leaking air. Now, if he's going to do the same thing for this medium-sized balloon, that means he's got to puff a little more often, right? And correspondingly, you'll have a same steady size that he maintains, but with, because he's got to puff more often. For the largest size, he has to puff quite often, right, in order to maintain this really high size. But each puff, remember, is the same size. What does this imply? What this means is that if you look at the jitters in these size graphs that I'm giving you, the jitters in the blue line, the size of the typical size of the jitters has to do with the size of the puff that the boy is blowing into the balloon, right? Which means that as the average size gets bigger, the relative jitter, right, it gets smaller with respect to the size. So, because the jitters here, they're the same size as the jitters here, but this thing is just a lot bigger. It'd be like if I gave you $10, $100, and $1,000, and I randomly added and subtracted $1 from your pile. If you only have $10, you're going to notice if I add and subtract $1 at a time, because you go from 9 to 11. It's a reasonable difference from time to time. If you have $1,000 and I add and subtract $1, you're not going to notice it nearly as much because you're going from $9.99 to $1,001. That's just a really small difference. That's the same principle in operation here. So the idea is that if this is how assembly works, then we should see a fundamental trade-off between the cell's ability to control small structures in the same way that it controls large structures. There's a trade-off. It's going to experience more noise assembly noise. It's going to have less control over small structures than it does have large structures. So what we can do then is we can go back to our microscopy data that we collected, and we can actually go ahead and measure the randomness. For aficionados in the audience, the randomness is just the standard deviation of the probability distribution divided by the mean of that distribution. You don't have to worry about that if that was too fast. Just think of it as a randomness parameter. So we can actually measure the degree of randomness for different sized organelles. And we can do this for a whole host of the organelles that I introduced in that cartoon a couple slides ago. So for a Golgi, the post office, for lipid droplets, the fat storage sites, peroxisomes, and mitochondria, which are energy burning sites. And our theory that we just went over with the balloon example would predict that as you increase the size, you're going to get a lower randomness parameter. That's why you get this negative sloping line. And this should be true for all of these different structures. All right, so that's what the theory, our own theory, we wrote this down and we wanted to test it. So then we actually go and measure, and we see nothing like that at all. In fact, what the cells have somehow been able to figure out how to do is they've been able to decouple their ability to control the size of the structure. So you can make big Golgi or small Golgi, for example. But this randomness parameter somehow stays flat. It's completely able to keep the two away from each other. Okay. So when my students Kianduk Panchanamiri and Issa Kalish came to me with these results, I said, okay, go do it again. We did something wrong. This is all measurement noise. They did it again. We did it 4 different ways. We used all kinds of microscopy techniques, including super resolution imaging. we kept getting the same answer. So then we said, okay, well, we better take this seriously then. And it was a puzzle, because I'm very used to dealing with theories that do this, and I don't know what to do with about a theory that does this. So we did what physicists often do when you're confronted with something like this. We drank a lot of coffee, and we stared a lot at computer screens and at paper, and finally we hit upon an idea that could allow us maybe to explain why it is that no matter what size the cell is able to grow these organelles to, it maintains this noise profile. Its ability to exert control stays the same. In order to understand this, let's go to our ball again, our boy blowing up a balloon again. Okay. So the theory that the model that we propose to explain this puzzle, it's related to the model we had before, with one really big modification. So you'll remember that for the small balloon, we said that the boy doesn't have to puff very often, because he just needs to maintain a small balloon size. But for medium and large, he needed to puff more often, because we assumed that that the air puffs were all the same size always, right? So what happens if we get rid of that assumption? What if instead we say that actually what the boy can do is actually puff at the same rate always, but instead allow the sizes of his puffs to change, in our case randomly actually even. So if you allow that, so now to blow the medium balloon, he takes the same number of puffs, he just lets the puffs get bigger. And if you think about it, when you blow a balloon, that's probably how you do it too. You're not sitting around, when you have to blow a big balloon, you're not going, like lots of times into a big balloon. You're taking big breaths in and blowing out a lot, right? So it kind of intuitively makes sense that a cell would do this. So if you allow the sizes of the puffs to get bigger, then you can get to these larger average sizes, but with larger jitters. And in fact, you can do the same thing for the largest balloon. You can have the same number of puffs, but you have even larger jitters. So you get to the same average, but the averages and the jitters, they go hand in hand now. So this is the idea we proposed to replace the sort of traditional model of how self-assembly works for organelles, to explain this puzzling data that we had. So how does it work? It's a nice story. It seems to fit at least one part of our experimental results, but we were not satisfied with just sort of coming up with a new idea post hoc and leaving it there. We wanted to really stringently test this model. So the way we did it is something that people who do work in my area called statistical physics do a lot of, which is we take our data, okay, and what I'm showing here now is real data from our recent paper on this, which is it's a depiction of the probability distribution of the sizes of these organelles, okay? And what we've done here is actually we've thrown all the different types of organelles onto the same plot. One thing to sort of appreciate about this is that if you put your traditional cell biology hat on now instead of your physics hat, it's crazy to put all these different organelles onto a same plot for any reason, actually. These organelles, the molecular mechanisms by which they're made inside cells, are vastly, vastly different from each other. There's no reason to expect that they have anything to do with each other, actually. The Golgi, for example, bud off of a different organelle called the endoplasmic reticulum. The peroxisome sometimes buds off from that same structure. Sometimes it fissions into two pieces. Lipid droplets actually do blow out like balloons from the endoplasmic reticulum also. So the mechanisms by which they're made, totally different from each other. And so we thought that a really stringent test of our model would be to say, okay, what if we take all this data from all these different organelles and see if we can't explain all these features collectively using our theory? Okay, so when we apply that random burst model that I just outlined in the previous slide, where the little boy is allowed to blow into the balloon with different sizes of puffs, this random burst model makes a very specific prediction about what all these data points will do if we squint at it just the right way. So if we, for example, divide out these data points by the average sizes of the puffs, and if we also then divide out the rate at which the boy puffs, if you do those two mathematical transformations on this data, our model makes a very specific prediction about what the data should look like. And it should look like this universal scaling curve that's shown on the right. So this black curve is a pure prediction from our model, and it has no what are called fitting parameters or anything. So there's nothing in here where we can kind of kind of mess with the data to make it and try to understand how it best lies on this data, on this curve, this black curve I'm showing. It's either going to lie on the curve or it won't. And that if it doesn't lie on this curve, our model is wrong. But of course, I wouldn't be telling you this if it didn't actually work quite well. So it turns out that if we do in fact do those defined mathematical transformations on this very scattery looking data from all these different types of organelles, and then apply them to this model that we just proposed, they very nicely fall on this universal scaling curve that to us is a very stringent test of this model's ability to predict cellular order creation out of this chaos. So this led us to conclude that in fact, this combination of simple model building and ideas from statistical physics, combined with very quantitative data, can actually tell us a lot about the assembly rules that cells follow in order to build these structures, to build order out of this chaos. All right. Are there any questions about, this is actually a pretty good point at which to take any questions if there are any. Mike, you're muted.
So there was a question early on,
Sure.
which maybe you could answer now.
Yeah.
Which is in the process of creating new cells, what's the lifespan of the parent cell?
Oh, that's a great question. Okay, so in the organism that we work with, which is budding yeast, this is the same yeast that we use to like make bread and beer and wine and stuff, right? So there's two types of aging in those cells. One is how many times it can divide before it dies, basically, and that's the one I think that is being asked about. And that's typically about I'd say, let's see, maybe about 20 to 30 cell divisions. Each one is about 2 hours, say. So they're about 40 to 60 hours before they stop dividing. That's for a single cell. And then, of course, there's this magical process, well, it's not magic, there's some scientific reason for it, but when a daughter cell is born, that clock somehow resets. This is actually a major area of research in aging biology, is how that clock resets when a daughter cell is created from the mother cell. Something that we don't know. Yeah, any other questions? I think I see a hand
one
up.
from Marty.
Yeah.
Yes, Dr. Mukherji, this could be a little fun fact, and perhaps your opportunity for 15 minutes of fame. And it has to do with a famous East Indian oncologist by the name of Mukherji, who you could be familiar with. Any relation?
Probably not. Oh, unless it's, well, no, probably not a relation, but if it's Siddhartha Mukherji at Columbia, then I do know him, but well, I know of him.
Okay, thank you.
All right. So, okay, so where we're at then is we have started to at least convince ourselves that we can, in fact, uncover some of these principles by which individual structures inside the cell are regulated in terms of their assembly processes, to see how it is that cells can create order out of chaos. Of course, compared to the cell as a whole, that's a relatively simple problem, right? Because not only does the cell have to regulate the assembly of each of those components individually, those components, they're not just operating in a vacuum, right? They interact with each other, they interact with the cellular milieu. So you can't, we will never be able to achieve a sort of holistic understanding of the cell if we just sort of look at them piece by piece like that. We have to understand something about the interactions between those components also in order to understand how the cell is able to collectively manage all its component parts to create stable patterns like growth. So that's, so this work that I'm going to show for the next couple minutes is much newer. It's much less refined, as you'll be able to tell by just the quality of the graphs. And so this is just to leave you with a notion of where we're going with this line of thinking in the future. So one way that, actually, let me go back. So one principle that is proposed to allow this coordination is to actually coordinate the pace of that assembly process, of all these various assembly processes, with these cellular scale phenomena like growth. Right, so it's a lot like this balloon animal, this Disney balloon. If any of you have been to Disneyland, you've definitely seen hundreds of these. So the idea here is that the delimiting membrane, that outer clear balloon, its rate of growth is going to somehow be coordinated with the rate of that blue Mickey Mouse balloon on the inside, right? Because if the Mickey Mouse balloon grows too fast, it's not going to allow that outer balloon, the cell as a whole, to function properly, right? They somehow have to be related to each other in order to maintain their proper balance. So for simple systems, there actually is quite a bit of data on how this coupling can actually happen. So my colleagues at Ethan Garner's lab at Harvard, they looked at this problem in the context of bacterial growth. What's shown here are individual enzymes from this bacterium that actually literally will deposit at the cell wall the new, basically, bricks that the cell wall is made out of. Okay, and the idea is that it just keeps depositing these bricks it sort of slides them in between the old bricks. And in that sliding process, it actually makes the cell grow outward. It sort of grows in this rod-like fashion. And I'll show you a little movie of how the enzyme's motion works. You'll see it's kind of circumferential, and it's basically depositing bricks all along the circumference of these cells. Let me try and turn this off. Okay, so here's this movie. So you can see that these little enzymes are moving round and round the outside of the cell, the outer boundary of the cell. And basically what it's doing along the way is depositing these bricks. They're called peptidoglycans for any aficionados in the audience. And basically that's, in this very simple system, how growth and self-assembly are coupled. Is literally you spatially restrict its activity to the location where its activity is needed. There's a similar example shown on the right here from my colleague Ahmed Yildiz at Berkeley. Here they're studying the assembly of a structure called the flagella, or the cilia. So the cilia, we have cilia, for example, in our lung airways and in our guts. These are these protrusions that are built by the cell. They come out, from the surface of the cell and sort of grow outward like little tubes. And the way these protrusions are built is you have literally little trains. They actually even call them trains in the literature that take the building blocks from the cell body, from the sort of bulk interior of the cell, and these trains will literally transport them out to the tips of the cilia leave them behind, and then come back. They'll actually even come back to pick up more building blocks to send them out. And this is how growth works. And here's a little movie of this. So you can actually follow these little fluorescent dots are actually individual trains that are actually going out to the tips of these cilia. Here's actually 2 cilia, and depositing these little building blocks. Okay, so now those are beautiful examples. I'm in awe of the kind of work that they've put into getting to take such high resolution, high accuracy videos of those processes. But of course, our playground that we've been dabbling in is even more complicated than that, right? We have not just one structure whose growth that we're looking at. It's not just the cell wall or the cilia, for example. We're actually trying to understand how this town is allocating resources to all these different parts of the town to manage its growth, right? That's how, if you were sitting in town hall in the nucleus and you were the mayor of this town, how would you decide, and let's say you wanted to grow this town to make it bigger, how would you decide where to send your resources? How much should you send to the power station to provide the fuel to actually power the rest of the town? How much would you send to the post office so that you can reliably send goods from one part of the town to the other? How much would you need to get rid of stuff too? How much would you put into the storage dump, right? These are the kinds of decisions that we think cells have to make in order to manage this really complicated assembly process at the cellular scale. So in order to actually answer those kinds of questions, we actually first needed to develop techniques to even be able to look at multiple components inside individual cells at the same time, right? Historically, when people try to look at many, many things inside the cell, I'm going to tell you right now that there are 4 distinct structures that are labeled in this micrograph shown before you. It's very hard to tell them apart because the palette of colors we have to work with when we actually label structures inside cells is very limited. The colors just run into each other. And so some people try to look at the shapes of these structures and distinguish them based on the shapes. Most people just give up after looking at 2 at a time. 2 is usually the most you can do. So we thought a little bit about ways that we could improve on that. And we started sort of exploring all kinds of modern statistical techniques that people use. It goes by various names. The one you probably have heard about the most is something called machine learning. And so what we've figured out how to do is we figured out how to leverage these machine learning techniques to actually take images that look like on the left, where all the colors are sort of mixed up, give it some more knowledge of what it should expect to see, and we can actually unmix all these mixed up colors in a machine learned image. And so now we have a really easy way of telling one structure from another based on the color, the precise color that it's lighting up with. So now we have similar cellular level data as we did in the previous example that we went through in detail, except now we have it for all these various structures inside the cell at the same time. What that lets us do is to map out these really high dimensional data sets. So our data sets are usually, something more like 12 or 24 dimensional. I don't know about you, I have a hard time with 3 dimensions. I can't do 4 or 5, never, and don't even talk about 24. So what I'm showing you here is a slice through that data set. It's a subset of that data that just depicts for every single individual cell, that's what each of these dots is, how many fat storage sites does it have? So how many lipid droplets does it have? How many power stations does it have? How many mitochondria does it have? How much of the lipid synthesis site does it have, this endoplasmic reticulum? And so for every single cell, we can actually pick off these properties of the cell. And now we can see not just how these individual pieces are working and what the control is on each individual piece, we can now actually see how the cell collectively emerges from all its component pieces. So for example, we can do all kinds of sort of modern data science type techniques on this data to actually understand the collective sort of interactions between all these different organelles that underlie things like its ability to grow. And we're literally scratching the surface of this kind of work. And this is these collective dynamics using these modern imaging techniques are where we're really excited to head next, to see if we can't figure out how the cell is able to coordinate overall growth with these collective interactions between its component parts. So that's what I wanted to share with you today, and what I want to leave you with, what you can tell your friends the next time you get to see them, is that a real millennial challenge for us as physicists and biologists is that we need to figure out how it is that cells are able to generate order and collective action out of these inherently unreliable parts, right? All the individual pieces are fluctuating like crazy because these are small systems. There's no brain in there. There's no that tells the molecules where to go. But somehow life is able to maintain some kind of order out of that chaos. And it's a real challenge to us to figure out how that's happening. But there's hope, right? Because we actually have the kinds of tools from statistical physics, from biology, and from sort of statistical tools from computer science and mathematics to actually make progress in this area. So I think it is an exciting time to try and answer these really fundamental questions in biophysics. So with that, I really want to make sure I thank my group for all the hard work they do. This is them. Every single person you see here contributed a ton to the work that I shared with you today. And I want to thank you for showing up.
I am virtually clapping in the way of Zoom.
That's great.
Exactly. Can I ask a question?
Absolutely.
Okay. So I'm wondering about whether or not there's a communication between various organelles through some sort of chemical messenger system that might also help with this.
So there's absolutely. Let me see if I can. I'll go back and. So there's absolutely all kinds of communication systems actually in between the different organelles. There's chemical messengers. So one way to think about this is that the outputs of given organelles, for example, the ER, this thing called the ER, it synthesizes all the lipids for the cell. The outputs of these are often inputs for other organelles, right? These other organelles cannot grow. They're bound by membranes, so they can't grow unless they've received lipids from this storage, from the synthesis site. So that's one way that these organelles interact. Another way that they interact is actually they literally are physically tethered to each other, and they transport material from one to another through these physical tethers. So my student Ali Ara, who came up with this image, she actually put these arrows in this diagram to indicate these physical tethers that she's actually studying. So she's actually using genetic tools to ablate these tethers, and then using our imaging tools to see what the consequence of that ablation is. Yeah.
Great. Thank you. Thank you. This was a fascinating talk. Thank you very much.
I have a question.
Sure.
Alice, and
Go ahead,
thank
Alice.
you. And I put it in the chat, but my question is kind of cosmic. Because these cells do respond to environmental influences in how they and how they grow and when they, divide and everything. Even with all these, I'm going to say, quote, mechanical mechanisms, could it be said on some level that cells have consciousness?
Yeah. I don't know. I think one thing that is definitely, there's a reason why I was attracted to statistical physics in particular. I mean, I sort of, when you're trying to figure out what to do, a lot of us who like physics, they sort of gravitate toward stuff that maybe Mike knows better than me, the stuff of particle, the stuff you read about when you're a kid, right? But this idea that that just breaking a system down to its component pieces will lead you to understand the system as a whole is difficult for me to see how it'll work for something as complicated as a cell.
Right.
And so I think a lot of the this idea of emergence and collective behaviors, it's a really nice system to study because whatever interactions lead to behaviors that we can't understand otherwise, we sort of ascribe to things like consciousness and stuff. But my guess is we'll probably end up getting to a place, maybe after a couple hundred years, where we get all these interactions down.
I had a question, and that is, you didn't mention what I generally think of as a major factor in control, and that's feedback. Especially negative feedback, which is sort of responsible for a lot of control in biological and physical systems also. And I don't know, is that sort of built into what you're talking about, or?
I am very glad that that you brought that up. So it's a very curious thing, right? Yeah, there's at molecular scale processes. So things like when genes are turned on or off, and how proteins are made, and how signaling systems work. There's a ton of feedback for sure. For some of these organelles at these mesoscopic, so at sort of larger scales compared to those molecular scale processes, there are definitely examples where there is clear feedback. So for example, the size of the nucleus in the cell scales very strongly with the size of the cell as a whole. So there's clearly some kind of communication there, probably some feedback control that appropriately scales those two at the same pace. There's these structures called the centrioles in the cell, which is where which organize the chromosomes upon the process of cell division, so that both daughter cells get this right number of chromosomes. There are always two of those, right? Because they're two daughter cells, and so those are under strong feedback for sure. And the mechanisms for that are still being worked out, but there's a lot known about it. For these other things, like the Golgi and the mitochondria and these peroxisomes, some of the other things you see in the slide now, it was very surprising to us that we were able to fit and explain the data we obtained using models that had no feedback in them at all. And I don't have a good explanation for why that is, because so my postdoctoral work, which I didn't get to talk about here, was about actually counting these objects. So how many, so today I talked about how big they get. In my postdoc, I worked about how many there are. And we actually wrote down a model for trying to describe how many there are. And we thought, okay, we're going to use this model to discover that there's actually feedback control, because the model will give us wrong predictions or something. But we never broke the model in that case. And so it's still a mystery to me why at this scale. It could just be it's really hard, because these are very complex assemblies made up of lots of pieces. And it might just be hard for the cell to establish a really strong feedback on these relatively large assemblies. I don't know.
What are some of the aspects of handling the time dimension of these processes? In economics, we have long run, short run, and just in time. I'm guessing that just in time is possibly a useful concept somewhere.
Actually, so half the time when I talk about this stuff, I talk about the cell as a town. Half the time I talk about it as an economy, actually. So I really like that analogy. And the idea of time is really important. It's actually something we don't have a lot of data on. That's actually one reason why we bothered to make these these spectrally resolved, color resolved imaging tools, because it lets us measure these things in time. You do see just-in-time kind of regulation in gene expression. So in the process of turning genes on and off, you'll often see that they'll only turn on upon a stimulus or upon the need for its products. That actually, Jacob and Monod, that is what they discovered. For these larger structures, again, I don't have a good answer. I'm clearly, when the cell goes from 1 to 2, it has to duplicate all these structures, right? And so it somehow has to coordinate in time how that duplication process happens. But we actually don't have a lot of movies of this process at this systems level scale. So that's what we're trying to get at now.
In economics, we have log jams.
Yeah, right.
These unreliable elements, nodules in the system. Thank you.
Yeah.
I have a question on the chat, but I can put it better talking. About aging, how much you can investigate with these methods? I mean, we know that we are less efficient. Is all the tissue less efficient? Are the cells that when they divide, I don't know, 20 times, they are less efficient? Are the new cells that come out of older tissue older than the ones from newer tissue?
Yeah, so that's a great question. It's actually one of the big application areas that we want to move some of this work into. So I'll give you a little story actually about aging with colleagues of mine at the Fred Hutchinson Cancer Research Institute wrote papers about this back in the mid 2010s. So it's definitely the case that aged cells have impaired function. I think that's not a big surprise to anybody. Mm-hmm. At this organelle scale, actually, some of those impairments have been identified. So for example, aged, and this is actually done in budding yeast cells, so I actually know this example pretty well. So in aged budding yeast cells, what the mother cell does is it retains pumps for protons, actually. So protons, of course, create the pH of the cell, right? Mm-hmm. It will retain damaged and old proton pumps and give the new ones to its daughter, okay? Mm-hmm. And what happens in that asymmetry is that the daughter is fine, right? It can regulate its pH just fine. In the mother cell, the aged mother cell, those impaired proton pumps result in an acidification of the bulk of the cell. And it's like, this is one of these follow your nose kind of stories. That acidification leads to impairments in the ability of mitochondria to function. And that causes a metabolic crisis eventually. And actually that's one of the major routes to age-related cell death in this organism. Mm-hmm. So the phenomena at this scale, at this organelle scale, seems to be critical for understanding that aging process. And there are proposals out to try and to sort of think about rejuvenation protocols to, for example, swap out old proton pumps from the lysosomal structure. So yeah, this is a really active area of research, actually. There are actually companies based on this kind of work, even.
Thank you.
Jeanette, did you have a question? You'll need to unmute.
Go ahead. Okay, in the chart that you showed, probability versus organelle size, which didn't match your predicted hypothesis, what is the probability of? What are we measuring the probability of? What are we looking at there?
It's the probability that a given cell will have an organelle of that size, or it's the probability that a given organelle will have that size. It depends on exactly which experiment we're looking at, but those are the two. It's the probability that it'll have that size.
So it's probability of size of whatever it is that you're measuring.
That's right. Yeah.
Okay. All right. Thank you.
Yep.
Okay, we have a couple of other questions in the chat. Lawrence Lewis, do you want to unmute and go ahead and ask the question that you ask, or do you want me to ask it?
Sure. So one of my questions was answered, And one of the questions actually that wasn't put in the chat is that when a cell divides, we're talking about the parent cell having issues with maintaining pH. But when a cell divides, how do we know which cell is which one is the parent cell and which one is the daughter cell?
That's a very good question, and it's very organism specific. So in cells like the ones we work with, so budding yeast cells, or actually in the majority, I want to say, of our own stem cells, so the cells that rejuvenate us, the division process is what's called asymmetric. So in budding yeast, for example, the daughter actually comes off as a little bud. Actually, if you remember back to that very first brass sculpture I showed, maybe I can even get back to it if I'm not slow. Yeah, this guy. So the process of cell division here is, it really is kind of like a balloon being blown up. So the daughter cell is the one that's basically starts out small and ends up as big as the mother. That's the daughter cell. And this process is asymmetric in that there is a defined site. These little circles here in this sculpture are indicated to show what are called bud scars. So when the daughter buds off of a mother, it actually leaves behind a scar on its cell wall. Stem cells actually work like this too, for the most part. Now, if instead I showed you experiments on bacteria or on a related organism called fission yeast, so this is called budding yeast because it buds. There's a fission yeast also that actually will grow out and divide in the middle. And there, the notion of daughter and mother is really, I think it's pretty arbitrary, actually. There's, you can try and track individual proteins to see, which ones were synthesized earlier, and if there's a cell that has more of those, then maybe that can be considered the mother. But I agree that there's this fundamental biological distinction between asymmetric and symmetrically dividing cells that plays a big role. And I think it has real evolutionary consequences too. People look at this a lot.
And David Cronin had a question. David, I think I unmuted you if you want to ask the question.
Yeah, so, well, in some ways you've answered. How has biology advanced as a result of your work, or possibly advanced? And vice versa for physics.
Yeah, I think that the big thing that we bring to this kind of work is an understanding of the sort of quantitative principles by which these biological systems are organized. We're not in the business of discovering the genes that underlie this process. Those have largely been discovered, actually, or the maybe mutations that underlie defects in these processes. What we really do is try and understand, when you put all these genes together and have their products interact, how do we even begin to think about whether that set of interactions is working appropriately or not? I think it's almost like a frame of mind that we bring to biology that people definitely have had. It's not that we're the first ones to think of things this way, but at this scale of cell biology and looking at, for example, organelles, there aren't a lot of examples of people just even looking at the question this way. In physics, I think that where we really contribute there is, it's a wonderful example of where non-equilibrium statistical physics is playing a central role in the operation of this system. So we're very used to thinking of, in statistical mechanics, usually what we think of are systems that whose probabilities kind of don't change with time, right? So they're in some sort of equilibrium. We teach this to our undergraduates. And we all know that a lot of phenomena, physical phenomena, don't obey those kinds of statistics because they actually are subject to inflows of energy and material. and dissipation of those two things. So getting rid of, heat actively, stuff like that. So I think these are really great examples that are of high biological relevance, but but have really important physical principles at play at the same time.
Okay, and I think we have a
strikes me then that in biology that they would discover some of their theoretical concepts are going to be found wanting as a result of your work.
I think that I forget if it was Don Cohn or someone asked about feedback, and when we were trying to publish my postdoctoral, for example, it was a very common question: is where's your feedback in your model? And I agree that even the idea that you should try to pursue the simplest possible explanation, quantitative explanation for the data that you have in hand, even that idea is something that biologists, because they are so used to dealing with dizzying complexity, often have, we need to talk about things like that, like why it's useful even to pursue the simplest possible idea rather than the idea that will for sure capture every single nuance of every single data set that you have. So I think that even philosophically, yeah, I'm hoping that this research sort of has a productive role in getting us to think about things the same way.
That's even melding equilibrium with dynamics.
Yeah. Yes.
I have a last question.
Well, go ahead.
Yeah, it might be a little bit lateral, but it really surprised me years ago knowing that we are a bag of collagen with cells in it. It surprised me recently that we have as many bacteria cells. Do those things, do the bacteria at least are organized in some way, or they're just passengers and making colonies there?
Yeah, this is definitely, you will find on our campus world-leading groups in just this area. It's actually partially that whole field is partially invented here by down at the medical school. So I'm going to say what I know about it, but certainly we have people on campus who are far more knowledgeable about this than I am. It seems that there are some organizing rules for where these bacteria are and which species there are. At the same time, the stability of those populations seems to be pretty low, in the sense that I remember a friend of mine who did experiments in this area showed that if you change your diet, like if you go from one locale and go somewhere else and start eating the food locally, the populations of the bacteria inside your gut will just switch overnight. I mean, literally overnight. So it seems to be very environment dependent also, but if you come to Mikhail Tikhonov's talk, I think he's speaking in this series, maybe even next time.
Yes.
Ask him. He knows a lot about this stuff. He works with these people.
Sure, thank you. Mm-hmm.
So
what about the collagen? Does it do anything, or it's just a support?
Yeah, I don't know. I mean, the going wisdom on collagen is it's just a structural filamentous component. Whether it actively signals or something, I don't know anything about that, actually. That's something I should look up.
Okay,
so one final question in the chat that hasn't been talked about, I think, it's humans have 5 senses. What are the senses of the cells?
That's a good question. Okay, clearly smell, and not smell in the sense that we know of it, but they are responsive to chemical inputs, like in terms of like the shape of the molecule will bind receptors and trigger events inside the cell. That's basically what smell is for us. We perceive it as smell because our brain then takes those signals and does all kinds of other things to it. But that's basically smell. Touch, for sure. My colleague Anders Carlson in our department works a lot in this area. And we have a lot of biologists in the biology department at WashU that work in this area, like Liz Haswell. If you touch the cell, it will open up ion channels in the cell's periphery that will allow fluxes of current through the cell, and cells respond to that. So that's basically what touch is. It's just flows of ions across membranes upon a mechanical stimulus. So cells definitely touch. I don't know, taste and smell, I'm going to call those the same thing. Hearing is something that there are obscure examples of acoustically active vesicles inside cells. They're actually being leveraged to use, people are actually deploying them in other cell types to actually try and acoustically image cells. So you can actually do ultrasound on cells, actually. There's a group at Caltech that, Mikhail Shapiro's group at Caltech does a lot of ultrasound on cells using these acoustically active vesicles. So maybe I'm sure there's got to be a reason they have those, but it's, as far as I know, not widespread. And sight, there is this crazy example that I saw recently where there's a particular kind of, I think it's cyanobacteria, so a precursor to chloroplasts in cells. They actually will arrange their membrane in such a way to act like a lens, and they will literally focus light onto a particular part of the cell so that they can follow light signals. So their cells can definitely see. I know of a very well worked out example of that. Again, not widespread. Our cells certainly can't do that, but it does happen.
Temperature.
Temperature. They definitely will respond to temperature, yes. So yeah, pretty much all the senses, actually, even single cells.
Okay, well, on behalf of our audience, I'd like to thank you for doing this.
Of course.
It's been great. And we will reconvene next Saturday. And, I think we'll have another great talk too.
Thank you for coming.
All for coming.
Thank you.